all for being here this morning. So today it is my pleasure to introduce our speaker, Dr. Juanita Merchant. Juanita joined the faculty at the University of Arizona College of Medicine in Tucson in 2018 as a professor of medicine in the UA Department of Medicine and chief of division of gastroenterology and hepatology, and is currently a member of the cancer biology research program, and is currently also serving as interim cancer center director for the University of Arizona Cancer Center. She is coming back home as she earned her MD PhD here at Yale School of Medicine, did her internship and residency in internal medicine at Boston, Massachusetts General Hospital before completing her gastroenterology fellowship at the University of California, Los Angeles. In 2008, Dr. Merchant was elected to the National Academy of Medicine and appointed a member of the National Institute of Health Council of Councils. And in 2016, she also joined the Board of Scientific Counselors for the National Institute of Diabetes and Digestive and Kidney Disease, a unit of the NIH. Prior to joining UA, she was on the faculty of University of Michigan. She is board certified in internal medicine and gastroenterology. She has written or co-written more than 165 peer-reviewed research publications and is editor or co-editor of 2 books and several book chapters. She is a co-PI on the NIH AG Forward program, which was developed to increase the number of academic gastroenterologists from underrepresented groups. Dr. Merchant has remained continuously funded by the NIH for her work in gastric and neuroendocrine tumors, head Hodge signaling, gastric cancer, and transcriptional control mechanisms in colon cancer. Please join me in welcoming Dr. Juanita Merchant.
Great. Thank you, Dr. Rogers.
Before
Oh,
you get started, would like to present to you this plaque in honor of your presentation, PDE inhibitors block MDSC metabolism in gastric endocarcinoma.
oh, great. Thank you. Okay.
So,
Oh, picture.
thank you.
Okay. Great. Well, I'm really excited to be here. I probably come back about once or twice a year. I'll be back in November for the Dean's Advisory Committee. But I'm excited to present to the Cancer Center today because I really love to get some feedback from the esteemed oncologists and faculty here at Yale. So with that, those of us, so as you know, I'm a practicing gastroenterologist, and for those of us on the diagnostic side, gastric adenocarcinoma is primarily initiated by an infectious organism, which I'll review on the next slide, and therefore is largely preventable. There obviously are some caveats, which we can talk about, particularly in underrepresented minorities. But here is the basic summary. Gastric cancer worldwide has, we used to say the second or third most frequent cancer, but it's now dropped to about the 5th, probably because more intensive, particularly in Asia, in terms of screening and surveillance. But still, if that type of diagnosis is still associated with a high mortality rate, about 27,000 cases, new cases will be identified in the US, and it's about 11,000 deaths. The important point here with respect to prevention is the infectious component that can initiate this cancer. And the person, Barry Marshall and Robin Warren, got the Nobel Prize for discovering the association of Helicobacter pylori, first with ulcers, but then made the association with gastric cancer. But also we need to think about dietary components such as high salt, nitrates. The other less frequent infection is a viral infection with Epstein-Barr virus or EBV. So in the US, the prevalence of gastric cancer has declined So probably from about the 1920s to about the 1960s, gastric cancer was probably the second most frequent cancer. But then with an improvement in sanitation, which has not occurred in certain places, being in Arizona, this is a big issue on the Native American reservations. But that was what was probably driving the decline in the US, because Helicobacter is found in the water table, basically. However, it is continuing to rise in minorities and immigrant communities. The other interesting issue with respect to gastric cancer, so that's really the distal cancer, and I don't remember if I think I do include a picture of the stomach for those not familiar about thinking about the different regions of the stomach, but the cancers that are arising in the cardia are rising and are thought to be more associated with the maybe increase in use of PPIs. I'll come back to that point in a little bit. But in general, the needle really has been moved more in Asia, where Helicobacter is pretty much endemic in the population. And certainly on the West Coast, where you see more of the Asian immigrants, it's still fairly prevalent with the first degree relatives. But you can see the highest incidence tends to be in East Asia, China, Japan, and Korea. Per capita, it's actually Korea is the highest. So what I'll be covering today is how we ended up starting to address this issue, or why we started looking at this question in terms of what is driving the inflammation to change the mucosa from chronic inflammation to the metaplastic changes in the stomach. And we came at this, or I came at this from the hedgehog signaling pathway, which I'll show you why in a few minutes. And then we started asking questions in terms of translating from our mouse models to what can we do in people. And in moving from Michigan to Arizona, have been fortunate to start to collaborate with some of the oncologists there to begin a phase 2 clinical trial, which we're very excited about based upon some of the findings in our mouse models. So again, from a gastroenterologist's perspective, what we typically are seeing, in many instances, patients really don't even come to be seen by the physician and already have metaplasia. So I scope many patients that just have chronic gastritis, sometimes metaplasia, but no, Helicobacter is nowhere to be found. What is the connection between metaplasia in the stomach and the esophagus? And when I looked into the history of this term metaplasia, what's interesting is that, so if people remember, you basically have that goblet cell that's normally in the small intestine, but it's showing up in the stomach or in the esophagus. So the pathologists like to say a normal cell in the wrong place, but that's signifying that the mucosa is starting to move more toward cancer. We think maybe more directly in response to the immune microenvironment, as opposed to the bug being there pushing the mucosa toward cancer. But this issue of metaplasia and the link of metaplasia to a cancer was really initially identified in the esophagus with Barrett's esophagus. So that metaplastic change in the esophagus is a precursor lesion, and we actually have surveillance approaches for patients that have Barrett's esophagus. So the debate in the GI field is, is it going to be worth to actually start doing surveillance for gastric cancer based upon identification of intestinal metaplasia? And the jury's still out. I'm actually going to an NCI think tank next week where we're going to be discussing this, whether we can change the recommendations for gastric metaplasia. But the reason why that is because the question becomes, who do we survey? When do we survey them? And how often, which is where the cost comes in. So this is a picture of, for those not familiar with, the 4 regions of the stomach. The cardia, which is where you see the incidence of this cardiac cancer is higher. And this was a nice review article by the gastroenterology group, Samir Gupta leading that from UCSD, where Cancer at the cardia tends to be higher in whites and less so in minorities, and is more strongly associated with GERD and obesity, and not with socioeconomic status. Whereas the traditional cancer that is associated with Helicobacter pylori infection tends to be in the body of the stomach where the parietal cells sit, and the antrum of the stomach, which is another name for it, is the endocrine part of the stomach, which is where the G cells that produce gastrin. The reason I'll be coming back to that in a second. And so it's this region here that then is connected to the small intestine. So this is more strongly associated with a decrease in socioeconomic status and underrepresented minorities. And so definitely in Arizona, we're seeing high incidence in Hispanics and the Native American population. So as I mentioned earlier, there's a increase in interest in the tumor microenvironment, which I think this is well known to the oncology group here, which is a very heterogeneous environment that is comprised of stromal cells, neuronal, endothelial, but most of the work now is really focused on the immune cells because this is the target for the checkpoint inhibitors. And so we're coming at this from the approach that if we can decipher a bit more about the tumor microenvironment, particularly in gastric cancer, where we already know that the initiation of the inflammation is from an infectious agent most of the time, that we can develop better targets for treatment and biomarkers. So this is actually an example of the what we call the Correa paradigm, which Pelea Correa, who is a epidemiologist who initially was in Colombia, South America, and made the observation and published in the Lancet in 1975, this observation that there was chronic inflammation in the stomach that progressed on to cancer. And this is looking at, obviously, this is an epidemiologic study looking at people over time, but he noticed that there was sort of this intermediate stage where some people had loss of the acid secreting portion of the stomach and a substitution of the normal epithelium of the stomach with this mucus phenotype, what we call metaplasia. And in humans, the pathologist will read out intestinal metaplasia, which is an example here, where you see goblet cells, even Paneth cells in the stomach. This is the intestinal type. The colonic type is actually more strongly associated with gastric cancer, and it's more this foamy type of mucus metaplasia that one sees in the stomach. And so there's about half the world is infected with Helicobacter and may develop this chronic gastritis, but then about 10% will go on to develop metaplasia and 1 to 3% gastric carcinoma. So the thought is that this the tipping point with respect to the progression toward the likelihood of progression toward cancer is at this step where there is atrophy and metaplasia. And we started asking the question whether hedgehog signaling might be important in this transition of the mucosa from chronic inflammation to the metaplastic change. And I've added here to emphasize that the Pelea Correa paradigm was basically formulated before the discovery of Helicobacter, and then once Helicobacter was identified, that the link was then made to this chronic and then atrophic gastritis. But we asked the question about hedgehog signaling, and really it was because of a incidental finding by Andy McMahon's group at Harvard in 2000, where they published a paper saying that the sonic hedgehog knockout mouse resulted in gastric metaplasia. It turned out that probably wasn't accurate. It probably was more hyperproliferation, but they didn't have any GI pathologists reviewing those slides. So, but based upon that, Mo El-Zatari, at the time, who was in my lab, we actually obtained the mice that are in which the LAC-Z reporter is knocked into the locus of the GLI1, which is the transcription factor and is the transcriptional readout for hedgehog signaling. So we obtained these mice. So the knock-in of the LAC-Z molecule, we're able to maintain these mice in the heterozygous or homozygous state, but essentially you have a total body knockout. And he infected those mice with Helicobacter. Now, we typically use Felis in the mice because you get a much more aggressive inflammatory response sooner. We were hoping to save a little bit of money and see changes, and not have to wait 6 months. But with pylori itself, using the human pathogen, it can take a lot longer, and the inflammatory response is not as robust. So he looked at the mice at the time of infection, 2 months after infection, and then 6 months after infection. And so you can actually identify fluorescently using an antibody. So in the uninfected mice, you see that the alpha smooth muscle positive cells, which are the myofibroblasts in the stomach, are positive for GLI1, and pretty much those were the only cells that were expressing GLI1. So again, GLI1 is in the stroma, and typically what happens is that the epithelial cells, such as the parietal cells, will make the ligand sonic hedgehog, and it's received by the stromal cells. So in the uninfected mice, it's the alpha smooth muscle positive cells, but after 2 months of infection with Helicobacter, not surprisingly, you have a pro-inflammatory situation where you have an infiltration of inflammatory cells, and those cells are positive for GLI1. And when we did flow cytometry, this is published several years ago now, mostly myeloid cells, but not T or B cells, were the cells that were expressing GLI1. And so just to summarize that, basically, we then were asking the question, well, what are these GLI1 positive immune cells doing? And what was interesting is that when we infected, again, wild type mice, and now what you see here is an immunofluorescent stain for the different cell populations in the corpus of, or the body of the stomach. Intrinsic factor in the mice mark the chief cells, whereas in human, it's normally in the parietal cells. HKTPase marks the parietal cells, and this GS2 lectin marks a mucus cell. So here in the mice that are infected for 6 months, you see that they're developing a metaplasia and atrophy. So this region should show parietal cells, which would be the orange stain, but you can see here they're kind of moved off to the side because they're starting to disappear and show atrophy and being replaced by this mucus phenotype. However, in the cells that, or in the mice that were heterozygous for the GLI1 deletion, or because of the LAC-Z insertion into the locus, or homozygous, they maintain the normal architecture of the stomach. And so we were really surprised by that because, as I mentioned earlier, it's the stromal cell that's expressing GLI1. So this was telling us right there that there was something going on in the microenvironment, in the immune environment, that was affecting the mucosa. So to try to summarize this quickly, so we started looking at hedgehog signaling in this transition from gastritis to metaplasia. I should also mention one of the issues with working with the mouse models is they never progress to dysplasia and cancer just with an infection from Helicobacter. So we could only look at this step. So I've just shown you that GLI1 is important in the formation of metaplasia. So this, like I said, is 2015, 16 we were doing this. So we did microarrays, and we identified this molecule schlafen-4. There certainly were quite a few other genes, but this one was interesting because there were some papers, so both schlafen-2 and 4, I should mention. But the reason why we didn't pursue the pathogenesis related to schlafen-2 is because 2 does not have a ortholog in humans. So there is an ortholog for Schlafen-4. So we wanted to eventually be able to translate the work that we were doing in mice into humans. So that's why we focus on Schlafen-4. So this was the further analysis of this locus, which we identified in the array of the mice, comparing wild type mice to the GLI1 knockout mice. You can see here that there's a decrease in Schlafen-4, which suggested that this gene was regulated by hedgehog signaling. And so we did chromatin immunoprecipitation at the time to determine that indeed Schlafen-4 is a direct target of GLI1. So you can show that it does sit on the promoter of Schlafen-4. However, I do want to get into in a second what exactly are these Schlafens. So the reason why we focused on them again is because there was a paper in Immunity in 1999 that said that these Schlafen molecules were involved in both T cell and myeloid cell differentiation. So that's why we thought, well, you know, if we're looking at hedgehog signaling and its role in the stroma and its effect in mediating the this gastritis to metaplastic transition, that would be a good target. So actually, I'm gonna just give you a quick primer. The Schlafen locus, however, is fairly complicated, and this is what I was kind of getting at. So we identified Schlafen-4. There's actually quite a bit of information from one group that's looking at Schlafen-2. We did see this go up in mice, but you can see that it doesn't have its ortholog in humans. So you'll hear me talk about, as we move to the human data, the ortholog for Schlafen-4, that's about 60% similar, is Schlafen-12L. So I'm just showing you this now, just plant that seed in your brain. These are what are called the intermediate Schlafens. And the reason why that's important is because the longer Schlafens, ones in green, have another domain that's a helicase domain that's thought to bind to nucleic acids. I will be coming back to this point later. Okay, so coming back to the mouse model, we, as I mentioned, we're interested in that gastritis to metaplastic change, and we've identified these immune cells that are Schlafen positive. And so to understand more of what they did, we created a very fancy mouse model. And I know some people are not as familiar with some of these, you know, kind of mouse tricks that we do. But essentially, we took the mouse promoter. It was a large back transgene. We hook it up to inducible Cre recombinase. We breed this mouse line to a reporter mouse line, TD tomato. So this hybrid mouse is expressing or can be expressed in the presence, when we give it tamoxifen, this reporter, so turning the cells red. But what we also did is to do a bone marrow transplant and put the bone marrow from these mice into irradiated mice, so that essentially only the immune cells are going to be labeled with TD tomato, and ask the question, can we lineage trace this Schlafen positive cell from the bone marrow of these mice that have recovered and infected with Helicobacter and waiting 4 to 6 months to see, you know, how they get to the stomach. Again, this is was published in 2016, but I just wanted to show you that it's really been a very powerful tool for us, because you can see here like stars in the sky. And what I'm showing you here is a wild type mouse infected with Helicobacter, but we're taking these mice at 4 months before we have seen the cells actually arrive in the stomach. However, if we breed those mice onto a background where the sonic hedgehog, the ligand signal, is goosed up, so it was pretty easy. It was just a PCMV sonic hedgehog transgene. We breed those mice, you know, with the TD tomato signal, and you can see at 4 months there are these cells that are TD tomato positive, Here's a high-power view. Since they're fluorescent, we can pull them out. You can see they have a granulocytic nucleus, and they are exhibiting markers of a granulocyte. Even better, you can certainly do all sorts of arrays, which I'll get into a little bit later. But more importantly, we could actually isolate these cells from the infected stomach and show that they had T cell suppressor activity. So we did the co-culture and show that they were really functionally myeloid-derived suppressor cells. So I wanted to show you, well, what's the connection between hedgehog and how this gene is regulated, and why I think I'm happy that we decided to kind of stick with this, even though nobody's heard of Schlafen. So what you see here is where you can isolate these cells from, we basically, create a pus situation by injecting them with thioglycolate, take the peritoneal cells, and then we can culture them and incubate them with recombinant sonic hedgehog, about a 5-fold induction of sonic hedgehog message, Helicobacter alone, 3-fold, but the two together synergize, but more importantly, interferon alpha, so type 1 interferons, 800-fold induction. This gene and that locus is very strongly induced by type 1 interferons. However, if you isolate those cells from a GLI1 null mouse, you can see that this is why this locus is still dependent upon hedgehog. You could get a little bit of induction, but essentially it's a dead promoter. So it's like you need 2 keys to unlock this gene and follow it. And we mapped so essentially the hedgehog signal, GLI1 is a constitutive signal. The inducible signal is through type 1 interferons. And so then we ask the question, well, in the infected mouse stomach, where is type 1 interferons coming from? And it turns out that, and we've done some later work that was published in 2022, but basically plasmacytoid dendritic cells are sort of resident dendritic cells that are the most, cell population that is probably sensing the debris field there chronically, and probably why it takes time for this to develop. So really putting this all together, and you may want to look at our gastro paper in 2022, what we're saying is that Helicobacter infection is detected not only by the epithelium, so the epithelial cells will also produce type 1 interferon, but sort of on a per cell basis, it's the plasmacytoid dendritic cell. There's a certain pathway with activation of the interferon response factors, which are the transcription factors that bind to the type 1 interferon promoters, releasing type 1 interferons that then will polarize what we now think is a neutrophil or granulocytic cell that has been sitting there and was recruited to the stomach at some point in time, but then this debris field and threshold must be reached over time. So these cells are PD-L1 positive, and we were able to show, as I mentioned earlier, that they do have T cell suppressor function. But analysis of these cells also reveals that they are producing other cytokines, not surprisingly, some of which that were of particular interest to us was IL-1 alpha and beta. And we think that, and that's why we think that it's the immune cells that are really picking up the baton and really pushing the mucosa more toward cancer as opposed to the bug itself. And recently, and I didn't put the reference in here, we actually had, for other reasons, had generated a triple transgenic mouse where we can inducibly overexpress IL-1 beta in the antrum. So you may ask, well, why would I bother to do that? And it's because the Helicobacter infection, whether it's Felis or pylori, when we infect the mouse, because the mouse stomach is actually a pH of 3 or 4 compared to our stomachs, which is pH of 1, the organism tends to only infect the corpus, not the antrum where traditionally you see it in people. So we really wanted to understand distal gastric cancer where we can drive a, much more aggressive tumor in the antrum of the stomach. And so we took we made a gastrin-Cree ERT2, crossed it to a TET activator, RTTA. So these are 3 different mice that have to be all bred together. So a lot of alleles. And then this mouse is then bred to a TET-on where we've inserted the IL-1 beta where it'll generate a secreted form. And so you give the mice tamoxifen. So the TET activator will sit in the cytoplasm until we give the mice doxycycline in the water. And so we keep them on doxycycline. And after about 6 months, about 40% of the mice will develop these ugly dysplastic looking tumors. I caution to call it cancer because the mouse models never metastasize. Even the colon, all the models that people talk about, they never metastasize. So, you can kind of quibble about what you want to call that. But I'll just say, you can see they're pretty ugly looking cells. And more importantly, these cells they do have and have recruited these Schlafen-4 positive MDSCs into the tumor. So at least we now have sort of a preclinical model to actually study. So going back again in time a little bit, so 2020, we started to do bulk RNA-seq, which we did with these mice that were TD tomato positive. What I want to point out here that was quite interesting, and coming back to the type 1 interferon theme, is that a lot of the genes that we identified, so this is the heat map, happen to be interferon, strongly interferon regulated, and were these guanylate binding proteins, or GTPases, GBP2, GVIN, and they're of the dynamine class of GTPases. This is our the changes in the heat map, the fold log fold change, but I am comparing it to a paper in 2019 where it was a really elegant study of both a mouse and human lung cancer. So there were 7 patients with lung cancer, and they had a mouse model using RAS, and I want to say P53, there was another gene where they were able to generate lung cancer. And they did a complete analysis by single cell sequencing of the tumor microenvironment. What they called N2 or neutrophil 2 cells, which we now are thinking those are those tumor associated neutrophils or TANs, they had the same gene profile that we identified in our Schlafen positive MDSCs. And I highlight here that their mouse N2 was positive for Schlafen-4. And here, this is the human counterpart for 7 patients. I think one of the problems we didn't see Schlafen-12L, but again, when you move to human, you've got a whole variety of stages, tumor types, et cetera. And so they did not observe it in that, but all the other genes were similar. We've also gone on to show that using proteomic analysis and using the Schlafen-4 antibody, that we can actually pull down and show that Schlafen-4, which I didn't mention, is actually a cytoplasmic, it's actually an ER membrane, endoplasmic reticular membrane protein. So I'll come back to that. So that even adds to the complexity. What are we dealing with? But interestingly, it forms a complex with, at least when we pull down, with many of these genes that we identified in the bulk RNA-seq. A little bit of a complicated slide here. Again, it's published for those that are interested. So if we take that pull down using Schlafen-4 antibody, and we wanted to know whether it had GTPase activity. So we take that complex where we've pulled it down and actually show that it can hydrolyze GTP. And so shown here, and it does that here, higher levels in blue of GTP, fold change, in the interferon treated cells where we do the pull down, versus we have also made recently a Schlafen 4 knockout mouse model. So if we isolate cells from those versus sildenafil. Now, why did I use sildenafil? I kind of skipped over that. And that's because some of the genes also were these cyclic GMP related phosphodiesterases. So we already were starting to think, well, maybe, there's already an off-the-shelf, oh, did I do that? There's already an off-the-shelf inhibitor of phosphodiesterases. Plus, I'm sure the oncologists are very familiar with, particularly from the multiple myeloma field, where you can use these phosphodiesterase 5-6 inhibitors as a sort of neoadjuvant. So that was one of the reasons why we thought, oh, let's see whether this works. And indeed, it also knocks down the ability of this complex to form GTP. So we put together this model, which I'm showing you here, that interferon will induce, because remember, it's a very strong inducer of Schlafen, so we can mark these cells. But along with Schlafen, there are other very important type 1 interferon regulated genes that appear to be somewhere in this pathway. And I try, this is this kind of a model, because essentially what these myeloid-derived suppressor cells, their ability to inhibit T cells has to do with them being able to gobble up L-arginine out of the environment so that the T cells can't proliferate. But what are these myeloid-derived suppressor cells are actually using that L-arginine themselves to, what I'm not showing here, generate reactive oxygen species. Here are some of the pathways. So arginase making nitric oxide, or NO2 making nitric oxide, which happens to be a cofactor for soluble guanylate cyclase. So guanylate cyclase generates cyclic GMP. Cyclic GMP, if it hangs around, is a cofactor for protein kinase G, which can, in some cell populations, trigger the cells to undergo cell death. So if you have high levels of something that's going to break down cyclic GMP, you're going to move the cells away from apoptosis, regenerate the sort of backbone for regenerating GTP. And so that's why we think, and I've shown you that Schlafen 4 is at least in a complex with these guanylate binding proteins, which need this GTP. So we think that there's a whole nother pathway or metabolism that pulls the substrate away from maintaining high levels of cyclic GMP. And you can essentially accelerate that, and we'll get back to that. Oops, going too fast. If we inhibit phosphodiesterases. So you can imagine if we block phosphodiesterases here, this is going to build up and you can trigger the cells to undergo apoptosis. So that's kind of the hypothesis that I want to keep in mind. Okay, so let's move on. We've moved to the next era where we're now using single cell sequencing. And I want to point out again that we're reinforcing what we initially observed. And I just want you to, this is published, but you can see here in our Go enrichment for, this is the spring plot. I won't bore you with all of that, but you'll notice that the Go enrichment, GTPase activity, GTP binding. So again, a lot of the genes, even in doing the single cell sequencing, seem to take us to these GTPase types of proteins. I want to highlight, though, this region here. which kind of didn't blow up quite as big as it should. But what we were kind of surprised about is that there's really 3 groups: low, medium high, and high expressors of schlafen. And this is what we're finding many times as you start to get into single cell sequencing, is that many of these cells exist in sort of different activation states. We haven't quite gotten to the pseudo trajectory. Somebody's working on that because you need a different program. But what you can kind of see is that the low expressors, group 3, which is this blue, actually it has more of the neutrophil genotype. So that would be, I guess, no. Anyway, I won't point it. I guess this group here. And whereas the higher expressing ones, there's one group, number 2, that tends to be, and so that's this cluster here, higher in nitric oxide 2, which is actually a different group than that express arginase. So this is just the mouse. So even that mouse cluster that we're already thinking that we're polarizing and becoming myeloid-derived suppressor cells from a granulocyte or neutrophil, they actually have different sort of activation states or different gene clusters that you can now identify by single cell sequencing. Okay, so I've given you a lot of information. So essentially, from the mouse model, what we're saying is that, and I didn't really give you the sort of how this all begins, but essentially when Helicobacter infects the stomach, it can, the dying parietal cells or injured parietal cells actually can release sonic hedgehog into the plasma. So some of the papers that I didn't talk about in detail, actually you can pick up sonic hedgehog in the plasma of the mice within 2 or 3 days. These cells track to the stomach, but the first 2 months or so of the infection, we're still in more of the pro-inflammatory stage. It's not till about, when we did a formal time course, about 5.5, 6 months of a Helicobacter infection in mice, do you actually see these cells actually generate enough interferon alpha in the tissue that, the reason why I'm crossing that out is that we actually infuse interferon antibody in our 2022 paper to show that we could actually block the polarization of these Schlafen-4 MDSCs, and we did not get, the SPIM is the term metaplasia that we use for the mice. It stands for spasmolytic polypeptide expressing metaplasia, but we just call it SPIM. Because in the mice, you actually don't see the goblet cells, so they had to come up with another way to market. And so again, what we're proposing is that if we block the phosphodiesterases and maybe these, along with the GTPases, that we can do the same thing. So what I've shown you is more in vitro data, but now I'm going to show you what it looks like with the knockout. So as I mentioned, this is a normal mouse. And like I said, we can goose up the whole signal and get the metaplastic change faster if we overexpress with sonic hedgehog. So the green staining you saw before is the metaplastic change in the mice. And when we do the conditional deletion, and we're deleting it using GLI1 Cre-ERT2. So we're deleting it in those myeloid cells that we originally identified the Schlafen cells in. And you can see that you start to read the normal architecture of the stomach. The parietal cells are shown here in white, are starting to come back. What about sildenafil? Didn't take much. We did 2 injections of sildenafil, same thing. And here I'm showing you an H&E where you can really see the parietal cells, which I'm used to looking at it, but these big pink cells are your parietal cells starting to return in the presence of just after 2 injections of sildenafil. And very recently, within the last couple of months, going back to our IL-1 overexpressing mice with those big ugly tumors. So here you can see in this low power view, here is the villi of the intestine. Here is the pylorus, the junction between the stomach or the antrum and the small intestine. These are Brunner's glands. Here, the tumors develop, and we're able to accelerate it if you give it the MNU nitrosamine. So instead of 40% of the mice alone, we get about 60% of the mice will develop these ugly dysplastic tumors. But 2 injections of sildenafil, we're able to melt those tumors down. And the reason why I put this in here, this is, again, kind of hot off the presses. I want to come back to, okay, I told you that schlafen is a ER protein. Well, guess what? It's an RNA binding protein. And so we actually have recently done a pull down, again, with the schlafen antibody. These are transfer RNAs. And what's very interesting is that it actually binds to very specifically, in an inducible manner, glycine and tyrosine specific transfer RNAs. I don't have time to get into it right now, but we can come back to it at the end. But I just wanted to start to close the loop of this is a very interesting protein, and why is it so important, and why an ER RNA binding protein is involved. Okay. I'm going to, because of time, I'm going to come back to this diagram, which I know is pretty complicated, because I wanted to show you our phase 2 clinical trial. So this is a collaboration with primarily a really talented junior faculty in oncology, Junaid Arshad. Rashna Shroff is our chief of Hem-Onc, and Aaron Scott are the trio of GI oncologists. And so when I presented this to them, Junaid suggested, well, why not just try, let's try see if we can set up a window trial. And so essentially, I didn't know what a window trial was, but he said, what we can do, because most of these patients are going to have to go to receive standard of care FLOT therapy and then go for a gastrectomy if they're stage 1 to 3. So we're only dealing with stage 1 to 3, well, 1B to 3. And so these are the window trial objectives. So the primary objective, and I didn't realize this, we can't just give patients tadalafil, even though the safety profile we know is pretty good. I'm not supposed to, I guess, because of CME, I'm not supposed to say the trade name. But anyway, so they're just focused on this feasibility and safety. But the secondary objectives shown here, to see whether there's some pathologic response. But my interest in what I'll show you, because the study is still ongoing, I'll just show you that of what we're looking at in terms of, does tadalafil in a patient with actual gastric cancer do anything, right? So remember, we've got to follow 12L. These are the exclusion criteria, study feasibility. So we've been going for about a year. We've got 6 patients enrolled, 2 patients have finished the study. But what I want to show, so our goal is to enroll 10 patients. When I moved to Arizona, I set up a repository for our endoscopy lab. So I or one of the other endoscopists will do if the patient is not referred in, if they're not referred in from the outside, that becomes a problem, because we actually want to try to do single cell sequencing at each of these intervals. So that really means that we have to do the endoscopy here. So just with the biopsies, jumbo biopsies, we can do single cell sequencing. And I just wanted to show you that even in a gastric cancer patient, we can stain for schlafen 12L. So it's there in the immune cells in the lamina propria of these tumors. So I'll just show you a little bit of, let's see. And I'm sorry, it ends up going counterclockwise because of the way the data gets uploaded into the cloud for the 10x genomic analysis. So what we were able to do, because obviously we run into problems with, we're able to capture the second interval endoscopy, so this one. But if the patient comes in from the outside, we basically do single cell sequencing. We have plenty of normal referrals to endoscopy that there's nothing there. They don't have gastritis. And so we can do single cell sequencing on those patients. So what I have circled here is the myeloid cluster in a normal patient. One of my patients that I had referred for endoscopy because I knew that I was having trouble eradicating Helicobacter. So they had Helicobacter gastritis. And here, intestinal metaplasia. And then this was one of the patients the first patient that was enrolled in the study. And so they actually have a lot more of these Schlafen 12L positive cells, which if you look at the just that gene, you can see here that this is where the myeloid cells are, but look at the normal gastritis, intestinal metaplasia. I'm sort of going in order. Sorry, I didn't give you the paradigm. But Schlafen 12L doesn't come on until you very strongly, maybe a little bit in the metaplastic stage, but until these patients you actually have gastric cancer. Now, you're going to say, well, what are these other cells? They're T cells. So this was the big surprise as we moved, and not surprisingly, when you move from mouse models to people, sometimes all bets are off. So we now also have to understand what's going on in these T cells, because you can see, again, Schlafen 12L is picked up in the T cells, in gastritis, and intestinal metaplasia. Now, I think I have one slide here. It turns out that in the actual cancer, the T cells that are most prominent that you don't see in the other groups are the exhausted T cells. So that's going to be a whole nother project to understand what is this molecule doing in terms of the metabolism of T cells. So we have our work cut out for us. I finally wanted to show you what happens with tadalafil. And so here's a cancer. So this was one of the patients where this was our first patient. So we didn't have, they were referred in from the outside. So we only had slides. And so this is the staining for CD11B myeloid marker and our Schlafen 12L co-localized here in the merge view, but here the high-powered view in the cancer, but with tadalafil, that we are eliminating these Schlafen positive MDSCs. Okay, so the takeaway, there seems to be overlap between the pathways regulating Schlafen 4, and we also believe 12L, and cyclic GMP dependent phosphodiesterases. And these inhibitors allow cyclic GMP to accumulate and induce MDSC apoptosis, and that's the mechanism. Their elimination, we think, we can at least see it in our mouse model. The big question will be, as we expand this trial and get past the safety stage, that this potentially may be a neoadjuvant for gastric. But again, these cells are in a lot of cancers, and we need to, think about it in several cancers. So that I just want to certainly acknowledge Lynn Ding, who moved with me from Michigan and has really carried out all of these studies. Acknowledge again our HEMONC GI group division and our Targets biorepository, and to thank the patients for their participation. Thank you. I'll take any questions. And yes. I don't know, there's also 2 questions online.
But my question is, the degree of expression in the myeloid cells, is it something innate or is it acquired? Do we know? Is it something that's hereditary tendency to have higher expression in certain individuals and lower in others?
So maybe our people
Uh,
should talk to this.
Yeah.
So your question is whether people are predisposed
Well, maybe
because they have SNPs or mutations?
right. I'm just saying, does it take like a two-hit phenomena where you have H. pylori infection, but there is innate overexpression of certain of these proteins, and that's when cancer happens. And I also had a second question. Do you think that some of the same pathways are involved in other types of gastric cancer, like smoking related or others?
So this pathway, I think, and is similar. I shouldn't, because of the type 1 interferon regulation, is it similar to like the STING C-gas pathway? I haven't looked to see where the parallel and the overlap is, but I would emphasize that, you know, DAMPs, but probably even PAMPs certainly can activate these plasmacytoid dendritic cells. The reason why I like focusing on the schlafen is because we're able to take it all the way down to the promoter, and we know why that promoter and those cells get marked. So it suggests that you really need a very strong induction of type 1 interferons, or maybe there's mutations in those IRFs, et cetera, constitutive. I mean, it gets pretty complicated where, you know, whether people may be predisposed or not. We are, some of the endpoints that we're looking at are, so TLR9, mainly because there is already information actually related to gastric cancer and Helicobacter that patients that have mutations in TLR9 may have a more aggressive response to an infection with Helicobacter. So that's, we're starting with more upstream. Yes.
Thank you. That was a great talk. So tadalafil is, prescribed for other things as well. Have you considered doing like a retrospective study and looking at, maybe stomach cancer versus people who've been prescribed tadalafil?
A retrospective study. Yeah, so in other words, it's in wide use. The problem is, and I think we're going to need AI to do these kinds of things, because it's really someone has to mine the clinical data. I'd have to see whether it's already out there. Most likely it's not for gastric cancer, maybe for one of the bigger cancers like lung or colon cancer. But I still think it's going to take some energy to pull it out of the clinical records and really analyze it. Really, offhand, I haven't seen any papers really looking at that, but it's an excellent question.
Thank you.
Clara.
Hello. Two questions. One, back to the inflammatory cytokine role, and you showed that overexpressing is sufficient to contribute. But if you sort of throw in inhibitors or utilize cell-specific deletion of IL-1 beta, TNF, you had a range of different cytokines. What's the effect of deletion in your model on the end outcome?
So deletion of like
TNF
TNF or
IL-1 beta.
we haven't.
Okay.
Yeah, we haven't gone there. You can imagine how many mice my mouse bill is out of control, and it's breeding all of those mice onto those backgrounds, which I haven't. That's why we did the antibody. It's a little cheaper.
And then I guess the second question is a little bit related to the spectrum of schlafen expression. If I know you mentioned in the mouse model that you don't see progression to cancer, it's more the metaplasia. But in human, if you try and sort of consider the transition between metaplasia to gastric cancer, well, I guess first, can you speak to some of the things that you think are contributing to that, enabling that transition in the 1 to 3% that sort of overlap with some of the pathways you've highlighted? In other words, you go the 10% have metaplasia, and then 1 to 3% actual gastric cancer, right? And so I guess in your studies that you're doing, where you're looking at, are you able to look at spectrum of schlafen expression in that subset that goes on to gastric cancer relative to those that stay in metaplasia?
Okay, so I'm trying to, so have we looked at, so you're taking gastric cancer, or you mean taking metaplasia, patients with metaplasia?
I mean, in the pathway, are you, and it can be Schlafen and it can be, for the full pathway in general. Are you able to see that those that progress to cancer are fit on the higher end of sort of, or on the altered end of expression of the pathway relative to those that remain in metaplasia?
Alter of, I'm so, I'm sorry.
Of your, can you segregate, like in any number of things, can you segregate the high versus low in the
Oh, oh,
Schlafen
oh.
pathway for those that progress versus that remain in metaplasia?
Oh, so, but in people or in the,
In people.
Oh,
In other words, it's
we
like
have,
from
okay.
bulk RNA or from other types of data sets that may have been done in the stomach.
It just really hasn't been done. We haven't really segregated the subtypes of Schlafen 12L cells in the patients at all. I mean, we're just, a lot of it is just numbers, and we're just happy to be able to grab, well, with the repository, the logistics is tricky, because I have to have our inpatient teams let us know there's a patient in house. Patient has to agree. Many times they don't come, or they come and we've already gotten the biopsies, and our hospital will not release that, even the tissue. So we have a lot of just sort of logistical issues, but I'll keep that in mind or I can send you the data. Yeah.
Data sets out there from the stomach, and then look at high versus lower expressors.
Yeah.
Ends of the.
I mean, I'll look, I just haven't come across it, but it's a good question. You've stumped me.
I may have missed this, but is persistent hedgehog signaling in the MDSC cells required to maintain the dysplastic tumors? And if so, is there a role for smooth end inhibitors?
Oh, Dr. Kaplan had raised that issue yesterday. We could think about vismodegib to revisit that. It's a good question. And I mean, I would try it out in our mouse models probably first. I do know that, so if you use a GLI1 null, we haven't used any hedgehog inhibitors, but basically as I showed you with the in vitro data, you need some kind of hedgehog signaling for that promoter to come on. The assumption is that the cells aren't or polarizing, but we haven't gone back to really explore that. Oh, there was this. Oh, okay. I thought there was 2 things on the line, but it was just the CME. Okay, no questions. All right.